Hi everyone, I hope you're well. And in this video, I'm going to be talking to you about gas exchange in different organisms. So historically, you've learnt mostly about mammalian gas exchange in humans. But today we're going to be talking about gas exchange in single-celled organisms. We're going to be talking about gas exchange in insects, gas exchange in fish, and gas exchange in the leaves of dicotyledonous plants. So the first thing we need to understand is all organisms need to exchange gases. They need to exchange gases because they need to take in oxygen for respiration, and they need to get rid of carbon dioxide, which is a waste product of respiration. So all organisms need to do this. We know this because respiration is a fundamental characteristic of life. Everybody does it. So everybody needs to access that oxygen. So some organisms, like single-celled organisms, can just access it directly from their environment because they're so tiny that their surface area to volume ratio is large enough to allow them to obtain enough oxygen to fit their metabolism. But larger organisms need to have specialized exchange surfaces. So any specialized exchange surface will share some certain characteristics. So we will see similarities between all specialized exchange surfaces. And you probably remember these from learning about them at GCSE. But all exchange surfaces will have a large surface area to increase the surface area to volume ratio, a short diffusion pathway. So most specialized exchange surfaces are one cell thick for that reason, to reduce the distance over which the gases have to travel. And they maintain in some way a steep concentration gradient. And there are a number of different ways that different organisms do this. All of these features increase the rate of diffusion. And that's constantly what organisms are trying to do. They're trying to get in enough oxygen to allow them to perform the amount of respiration that they need to do for their metabolism, to get enough energy to do all the things they need to do, and to allow them to get rid of any waste carbon dioxide. And that's really important because we can't have carbon dioxide building up in our bodies, because carbon dioxide is toxic in large quantities. Carbon dioxide will dissolve in tissues as carbonic acid, lowering the pH of tissues, and we know the problems that arise from lowering the pH. We know that this can impact on enzymes and it can cause them to denature. So it's really important that we get rid of this excess carbon dioxide. Single-celled organisms are really amazing creatures. When we talk about single-celled organisms, we could be talking about bacteria, prokaryotes, we could be talking about protists, and we could be talking about single-celled fungi like yeast. So there are lots of different kingdoms that contain single-celled organisms. So there are many different examples of single-celled organisms, and they're utterly beautiful and massively diverse in their structure. And I highly recommend you just looking up some beautiful images of single-celled organisms. But these are able to absorb and release gases by diffusion, simple diffusion across their outer surface. This is because they have a large enough surface area to volume ratio for gas exchange to occur across, to suit their metabolic needs. They also have a thin surface. Now I just want to this is slightly odd wording here, a thin surface. So when we talk about a thin surface, we're talking about their cell membrane. Now there's no such thing as a thin or a thick cell membrane. All cell membranes are exactly the same thickness. They're all 7 nanometers in diameter. And we know this because each phospholipid, and cell surface membrane is a phospholipid bilayer, each phospholipid is 3.5 nanometers long. So this thin surface, this cell membrane, gives a very short diffusion pathway for efficient and fast exchange of gases. Plus, single-celled organisms don't have a very high metabolic rate. So they don't need an enormous amount of oxygen to fulfill their needs. So single-celled organisms do not have a specialized gaseous exchange surface because they're tiny enough that the distance between the outside world and their center of their body, which is the distance that matters, is really, really small. And so gases can just diffuse freely across their surface. Now, when we move on to multicellular organisms, this is obviously different because the distance between the outside world and the center of their body is much greater. So they need to develop specialized gas exchange surfaces, essentially to enable each cell of their body to have access to the outside world. We're going to start off by talking about insects, and this is a beautiful image of a caterpillar here. Can you see these tiny little holes all along this body here? These are called spiracles. And these spiracles are going to be important later when we think about the gas exchange system of insects. As our first example, let's have a look at this grasshopper. So think back to those spiracles you saw on the photograph earlier. These spiracles sit on the outer surface of the insect, and you can see here on this diagram the spiracle openings. It's not so easy to see, but they're all along the surface of the insect, so they would just be everywhere on the surface, okay? Now, these spiracles are holes, and these lead down into thin tubes that lead right down into the center of the insect's body. So there's an extensive network of tubes that lead into the insect's body from the spiracles. These tubes are called tracheae. At the ends of these tracheae, and we'll look in detail in a minute at the ends of the tracheae, but at the ends of these tracheae there are closed-ended tunnels, tiny air sacs where gas exchange takes place. So the insects have tiny pores on their surface, which are called spiracles. Air moves through these into tiny microscopic air-filled pipes, which are called tracheae, and these branch into smaller dead-end tubes that are called tracheoles. These tracheoles have permeable, thin walls, so this is going to be the site of gas exchange. This enables oxygen to diffuse directly into respiring cells. So at the tracheoles, oxygen travels down the concentration gradient towards the insect cells, and carbon dioxide moves down the concentration gradient towards the spiracles to get released into the atmosphere. So let's look in a little bit more detail at what's going on inside the spiracles. Okay, so take a look here at this diagram. So here you can imagine this is the skin of the insect and this is the spiracle. Okay, and then down, so the spiracle is the open pore, and then the air flows down the tracheae, so this open-ended tube, and then it gets down towards these tracheoles. Okay, these tracheoles are the dead-ended tubes that lead down into the muscle cells of the insect. So we've gone down from the outer surface of the muscle, from the exoskeleton of the muscle, down into the muscle tissue of the insect down here. So the oxygen-rich air is traveling down here. Now, when it gets down here, this can then just diffuse directly down its concentration gradient into the cells of the insect, and the cells of the insect surround the tracheoles. Okay, so they're just sort of everywhere because the tracheoles branch into every part of the insect's body. Notice that the ends of the tracheoles are filled with fluid. This is really important because this enables them to change their rate of gas exchange. So we've got a couple of things that we see in this diagram that enable them to change their rate of gas exchange. We're going to talk about both of them. The first is the muscles. So these muscle cells rhythmically contract and relax. So as these abdominal muscles rhythmically contract and relax, they physically force air in and out of the spiracles. This is essentially exactly the same way as you breathe in and out. Your diaphragm and intercostal muscles contract and relax to force air in and out of your lungs. And the abdominal muscles of the insect rhythmically contract to force air in and out of the spiracles, and the rate of contraction can change to change the rate of gas exchange, to change the rate of exchange of gases in the tracheae. The second thing is the fluid. So, if these cells that are surrounding the tracheoles start to not have access to enough oxygen, so the rate of gas exchange is not fast enough, they will be forced to start undergoing anaerobic respiration. When they undergo anaerobic respiration, they will produce lactic acid. The lactic acid will lower the water potential inside these cells. Therefore, water will move from inside the tracheoles into the cells down its water potential gradient. That means that the level of water in the tracheoles decreases, increasing the area available within the tracheole for air. And now there's more area available for air to enter the tracheole, and the rate of gas exchange increases. As the rate of gas exchange increases, the cells surrounding the tracheole have access to more oxygen. They do less and less anaerobic respiration. Their water potential increases, and then water returns back into the tracheole, and the rate of diffusion of gases decreases again. So this is a way that they can modify their gas exchange rate to ensure that it satisfies the metabolic needs of the insect at any time. So some key ideas there. First of all, the new term, spiracle tracheae, tracheoles, and then these two new key ideas. So the rhythmical contraction of the abdominal muscles for gas exchange, and also the role of the fluid in changing the rate of gas exchange. So the other thing that insects have to contend with is excess water loss. They don't want to dehydrate. And exchanging gases tends to make the insect lose water. So they have evolved adaptations to minimize water loss. Why does exchanging gases tend to make the insect lose water? Well, it's an open space into the atmosphere, and so water will evaporate off the surface of the tracheae and the spiracles out of the body of the insect, and so they will dehydrate. So if an insect loses too much water, they can close their spiracles. So they can use their muscles in their abdomen to close their spiracles. This will prevent excess water loss. Now, that might sound pretty stressful, like somebody closing their mouth and their nose to prevent water loss, but not getting enough oxygen. But actually, insects don't have to close all of their spiracles, and they've got many hundreds of spiracles all over their body. So they can close some of their spiracles to reduce water loss, therefore not reducing gas exchange to zero. And also the metabolic rates of insects is often quite low, so they can cope for a little bit longer than we could with a lot less oxygen. Insects are also covered in a waterproof waxy cuticle. And I know that you're thinking, oh, I've heard about these waxy cuticles before. That's like on plants. Well, although it might be made of a slightly different substance, it serves exactly the same purpose as the waxy cuticles that we see on plants. It prevents the evaporation of water. Insects also have tiny hairs around their spiracles to help reduce evaporation. So let's take a look at some of these tiny hairs in the spiracles. So we're just going to have a look at a couple of scanning electron micrograph images. So this one's really beautiful. We can see the scanning electron micrograph image, and we can see the spiracle, the spore, on the surface of the insect skin, and then we can see all these tiny hairs on the inner lining of the spiracle. You can also see all these tiny hairs on the surface of the skin of the insect. Take a look at this one. So you can see the spiracle here. And you can see how these hairs, you can see how if water was to evaporate out of this spiracle, it would just get trapped by the hair and it would recondense and fall back into the insect's body. And I think this one is a really beautiful one from the surface of a caterpillar, because you can see, first of all, the spiracle is sunken in a pit, just like you learned about in xerophytes in plants. The stomata are sunken in pits to reduce water loss, and you can see the spiracle, the pore on the surface, and then you can see all these hairs here, each one. So if a little bit of water evaporates, it'll get trapped by these hairs and then recondense and prevent it from evaporating out away from the insect. But you can also see this extensive network of hairs or spikes on the surface of this caterpillar. And last one, just because I think it's really beautiful, so this one's got a little bit of added colour in this scanning electron micrograph, and this is also a caterpillar, and I think it's really beautiful the way you can see these hairs sort of branching out and protecting the whole of the spiracle so that gas exchange can occur, but water loss is massively reduced because any water vapour that's trying to leave will recondense on the surface of these hairs and then be trapped in the insect's body. Okay, so that's how insects control water loss when they're also doing gas exchange. The next one that we are going to look at is fish. So let's take a look at this diagram. So when fish do gas exchange, they open their mouth and water flows in through their mouth. And then at the back of their mouth, there is a flap called an operculum. So when you do your fish dissection, which you will do as part of the A-level, or, if you just order fish from the butchers, that's a really good idea as well, just to get an idea of it. Just have a look at a fish head. You can pull back at the side of the fish head and you see this operculum. So I think this image might help you to visualize it. So you can see this sort of rigid flap that's being held up here, and you can kind of see, if you look through it, the open mouth of the fish. So like here's the fish mouth over here, and then this is the rigid flap. And then these are the fish gills. Okay, so these are the gills. So water will flow through the mouth and then over the gills and then out. This is a little bit more of a slightly gruesome image, but I think it's quite a good one. So just have a look here. You can see what somebody has done is they've literally put their hands in each of the operculum on either side of the head of the fish and pulled it out so you can really clearly see the fish gills. So you can see the fish gills here, and you can see how they're held in place in the fish's body. And we're going to be looking at the ultrastructure of the fish gill, but you can already see this lovely sort of angle at the base of the fish gill down here. So hopefully that's helped you to visualize it a little bit better. So the water flows through the fish's mouth and then out of the operculum, and as it does so, it goes across the fish gills. So let's just have a look at the fish gills in a little bit more detail. So here we can see the fish gills have the gill arch at the back, which supplies the gills with blood vessels. And then if we were to look at the gills themselves in a little bit more detail, we would see that we have capillaries running into the gill arches, which are oxygen poor. So we're providing the gills with low oxygen blood, which is exactly what you remember from learning about the lungs at GCSE. We provide our lungs with low oxygen blood. And you've got higher oxygen concentration in the water. So the gill lamellae is the site of gas exchange. So this extension of the gill membrane is where exchange occurs. So the membrane of the gill lamellae is very, very close to the capillaries, reducing the diffusion distance for gas exchange. So gases are being exchanged from the gill lamellae into the capillaries and in the opposite direction. So you have oxygen poor blood coming in, and then you have oxygen rich blood going out. So it's just like the lungs really, except instead of having these sacs, these alveoli, we have these sort of flattened membranes instead, split first into gill filaments and then further increasing their surface area by each gill filament being covered in these extensions called gill lamellae. And it's the gill lamellae that are probably the most analogous to the alveoli, and they're the site of gas exchange. So a few new key terms there that you need to know: the gill arch, the gill filaments, and the gill lamellae. So it's also really important that we understand the structure here, that the lamellae are surrounded by these capillaries for gas exchange. So the gill lamellae are thin, and they have a huge surface area to increase the surface area to volume ratio of the gills, to increase the rate of diffusion. They also have a really good blood supply. So they're covered in capillaries to maintain the steep concentration gradient. The next thing we're going to talk about is a bit more detail about how they maintain this steep concentration gradient between the water and the bloodstream. The water and the blood So the water and the blood travel in opposite directions. So let's just think about that for a second. So we can see that the water is traveling across the gill lamellae in this direction, and the blood is traveling in this direction. So the water and the blood are traveling in opposite directions. Water, blood. This means that as the water flows across the gill lamellae, there is always a higher concentration of oxygen in the water in comparison to the blood. So the blood that first arrives and is travelling across, let's just imagine that had 100% oxygen concentration. This is meeting blood that's been travelling through the gill lamellae and has been being oxygenated. So it's going to have a high oxygen concentration, let's say about 80, but not as high as the water. So a little bit of oxygen is removed from the water as the oxygen diffuses from the water to the blood down its concentration gradient across the membrane of the lamellae. Then a little bit further on, so we've got a lower concentration of oxygen in the water, but it's still higher than that in the bloodstream. A bit further on, a lower concentration of oxygen in the water, but still a bit higher than in the bloodstream. And as we get right to the end here of the gill lamellae, we've got quite a low concentration of oxygen left in the water. So here it's about 15%, but that's still higher than the 5% that we see in the bloodstream. So we're still going to have diffusion of oxygen into the bloodstream. This is known as a countercurrent flow, a countercurrent exchange. This is a really important term, and you'll actually come across it in a few key areas in biology. So it's countercurrent because they're moving in opposite directions, and the effect of them moving in opposite directions increases the rate of diffusion. This is because the water that is coming into contact with the lamellae is always going to have a higher concentration of oxygen than the bloodstream, and therefore diffusion of oxygen will always be in the direction of the blood, and diffusion of carbon dioxide will always be in the direction of the water. So let's just take a look at that concept in a graphical form. So this is a really important graph. So let's just take a look at it here. So here on the x-axis we've got distance along the gill plate, and on the y-axis we've got percentage saturation of oxygen, and the red represents the blood and the blue represents the water. So when we first arrive at the gill plate, the water has a very high oxygen saturation, and so does the blood, but it's lower. So the oxygen is going to diffuse from the water to the blood. To maintain a large or a high concentration gradient between the water and the blood, which speeds up the rate of diffusion. So this is a really important idea. So we can see then how fish gills satisfy the need for a high gas exchange rate to satisfy their metabolism. They have a large surface area to volume ratio, enabled by the gill filaments and the gill lamellae. They have a short diffusion distance between the bloodstream and between the water, and they maintain a steep concentration gradient with a good blood supply, with a countercurrent exchange system, with the blood travelling in the opposite direction to the water. So all the oxygen that's dissolved in the water can be absorbed into the bloodstream of the fish and then transported around their body. Okay, so the next organism that we're going to look at for its gas exchange system is the Dicotyledonous plants. Now, don't worry if you hadn't come across that word dicot. All that's really referring to is any flowering plant that has two leaves in the embryo of the seed. So two tiny leaves or cotyledons in the embryo of the seed. And it's really just most flowering plants that you've ever heard of. So let's just take a minute to have a look at this light microscope image of the leaf of a dicotyledonous plant. So we know that the leaves are the site of gas exchange. Okay, so we can see the upper epidermis here, not labelled on here, but present is the waxy cuticle. So we've got the waxy cuticle here, and the waxy cuticle is important for preventing evaporation of water from the upper surface of the leaf. And then below the upper epidermis, we've got the palisade mesophyll cell, and we know the palisade mesophyll cells, these are stacked cells, jam-packed, full of chloroplast, and they're the main site of photosynthesis. We can see that we've got the vascular bundle here with the xylem and the phloem. And we know the xylem carries water around the plant, and the phloem carries dissolved sugars from photosynthesis around the plant. And then now we see the spongy mesophyll. The reason it's called the spongy mesophyll is that there are lots and lots of air spaces, and these air spaces are the site of gas exchange. Now, how do gases actually enter and leave the plant leaf? They leave through the stoma. So the stomata are tiny holes on the underside of the leaf, and the opening and closing of the stomata are controlled by these cells here, these guard cells. So I'm just going to show you a quick picture of a stomata on the underside of the leaf. So this is an absolutely beautiful electron micrograph image. So in here we can see the stoma, okay, so we can see the pore through which gases are exchanged and through which water is lost. And we can see these kidney bean shaped guard cells either side. We can also see some of the cells of the lower epidermis on the underside of the leaf. This is a scanning electron micrograph image. So let's talk about the process of gas exchange then in these dicotyledonous leaves. We're going to use this image. It's very similar to the other one, it's just in this one you can see the stoma a little bit more clearly and you can see that it's open. So we're going to use this one. So gases are exchanged between the cells of the spongy mesophyll across these membranes and into the air spaces here inside the leaf. From these air spaces, the oxygen can then travel out of the leaf via diffusion, and the carbon dioxide can travel into the leaf by diffusion. So we know that plants in general make an excess of oxygen because their photosynthetic rate is during the day usually higher than their respiratory rate. Their respiratory rate is obviously constant, so at night their respiratory rate is higher than their photosynthetic rate because at night they don't do any photosynthesis because there's no light for photosynthesis. But in general they're making an excess of oxygen which they then release into the atmosphere, and they take in carbon dioxide because carbon dioxide is a substrate that they require for photosynthesis. So through the stoma they can also lose water vapor, and that's why the stomata can open and close, and these opening and closing of the stomata is controlled by the guard cells. So if we think about the guard cells for a minute, we know they form this kidney bean shape either side of the stoma. So in guard cells, there's a thicker cell wall laid down in the middle, so they don't expand equally when they gain water. So when water enters the guard cells, they expand in a way that causes them to bow outwards, and this creates the stoma. When they lose water and they return sort of to this shape here, the stoma closes. So the plant can control the opening and closing of the stoma through the entry and exit of water into the guard cells. So, how are dicotyledonous plants adapted for gas exchange? They have a really large surface area to volume ratio. They have mesophyll cells that have air spaces. Now remember, gas exchange actually occurs between the mesophyll cell membranes and the air spaces. When we talk about the large surface area, that's what we're talking about. We're talking about the large surface area of the mesophyll cells exposed to the air spaces. It's easy to get this confused with when you're talking about the large surface area of leaves in response to the size of the whole leaf when they're trying to absorb light for photosynthesis. So when we talk about large surface area, we're specifically referring to the exposure of surface of the mesophyll cells to the air spaces on the underside of the leaf. Okay, so it's really important that you understand that point. That's one that students often get a little bit confused about. And they have these stomata, so they can change the number of stomata. So we would expect plants that do a larger amount of gas exchange to have more stomata. So these are pores that on the underside of the leaf, on the lower epidermis, which can open and close to allow the exchange of gases. And then you've got guard cells, and we know that guard cells control the opening and the closing of stomata, and they do that by gaining or losing water. So just like insects, plants have to contend with controlling excess water loss because they don't want to dehydrate, and plants will wilt if they dehydrate. So it's really important because they rely on water in their xylem, they rely on water in their stem in order to remain upright, in order for their leaves to remain broad and flat so that they can absorb light for photosynthesis. So it's really important they don't lose excess water. So plants can open and close their stomata in order to minimize water loss. So stomata are generally open during the day for gaseous exchange. So water enters the guard cells, making them turgid, which causes the stomatal pore to open. But if they begin to dehydrate, the guard cells will lose water and become flaccid. This closes the stomatal pore. So often plants will have closed stomata in the very middle middle of the day, which might seem a bit counterintuitive because you're like, well, in the very middle of the day, you've got the most light, you need the most carbon dioxide to be doing the most photosynthesis. But it's better to do a bit less photosynthesis and not die of dehydration. So they're constantly making that trade-off. Another way in which plants control their water loss is by having a waxy cuticle. So they have a waxy cuticle on their upper epidermis to prevent water loss from their upper side of their leaf, which is obviously, without the waxy cuticle, would be the side of the leaf that would have the most water loss because it's exposed to sunlight. Some plants live in very dry, arid environments, and they need to have special adaptations to protect them from losing too much water and dehydrating and therefore wilting. These plants are called xerophytes. So we're just going to spend a minute talking about the special adaptations of xerophytes to prevent water loss. So xerophytes are plants which are specially adapted to live in dry or windy habitats. And I'm just going to talk about windy for a minute there. Windy, because in windy habitats, any water that evaporates will quickly get blown away from the leaf, and therefore you'll have a steep concentration gradient for water between the inside and the outside of the leaf, and so the rate of transpiration will be really high. So in plants like this, they're always trying to reduce the rate of transpiration, the rate of evaporation of water from the stomata in the leaf. So how are they adapted? Often they will have sunken stomata, so stomata in these little pits. This creates a tiny microenvironment. So say a little bit of water does evaporate out of the stomata, it just gets trapped in this space. It hits the walls, it recondenses, and it creates a tiny, humid microenvironment. So it traps moist air. This reduces the water potential gradient between the inside of the leaf and the air, and therefore the rate of evaporation of water slows, so the rate of transpiration slows. They also have hairs on their surface. This is just like we were talking about with the insects. These hairs, again, trap moist air, again, reducing the water potential gradient because the moist air will recondense. It will create a humid microenvironment, and there will be a little or no concentration gradient between the inside of the leaf and the air, and so the rate of transpiration will slow or stop. The other thing they can do is they can curl their leaves around, and therefore we see them as spikes. This acts to trap a little microenvironment here of still air. Because the air is still, any water that evaporates out into this air doesn't blow away. It doesn't get lost. And so you create a very humid little microenvironment. So we say the air becomes saturated with water. So the air has a really high water potential, and so there's no water potential gradient, and so there'll be little or no transpiration of water out of the stomata into this airspace. Also, and I mean a very obvious one, is they can reduce the number of stomata because this will always prevent water loss. They also can have thick waxy cuticles. So they could increase the thickness of their waxy cuticle in comparison to non-xerophytic plants. And they could also have that waxy cuticle on their stems as well as on the upper side of their leaves. Also, xerophytes will sometimes have this thicker waxy cuticle on the lower epidermis of their leaves as well as the upper to help reduce water loss. So there are a number of things that xerophytes do to reduce water loss. Okay, so in this video, we have actually covered a lot of information. We've covered gas exchange in single-celled organisms, gas exchange in insects, gas exchange in fish, and gas exchange in dicotyledonous plants. So we've also talked about some of the problems that these organisms face as a result of gas exchange, so how they protect themselves from excess water loss. So you need to go away and you need to learn the terminology, so how you could label any diagram, how you could use these words to describe the process, and how you could explain how gas exchange occurs and why these structures look the way that they do. But well done for concentrating, and I will speak to you soon.